Welcome everyone. While many of you know me, those of you that may not, I'm Kerri Kaplan, president of the Lustgarten Foundation, and it's my pleasure to welcome you to our year-end research update. So I just want to say, while Lustgarten has had some challenges this year with so many fundraising events going virtual, I'm actually really happy to report that we're still looking at a really strong year fundraising wise, and we've not had to cut any research programs. We've not cut any research funding. As a matter of fact, we've continued to launch new ones that we had talked about at the beginning of the year, and to meet this whole year and launch important programs. That's what you're going to hear about today. That research is not at all stalled. It's too important, pancreatic cancer research. So we've really continued at a fast pace. And of course, we couldn't have done that without all of you. I'm truly overwhelmed by the amount of support that our community has shown us this year. And for that, I really want to say on behalf of our staff and the board at the Lustgarten Foundation and patients everywhere, thank you. I do want to, though, of course, thank today's sponsor of our program, Ipsen. Ipsen has been a wonderful partner of the Lustgarten Foundation and really jumped at the chance to sponsor today's event. I want to leave the most time for our panelists, so it's my pleasure to now turn it over to Dr. Andy Rakeman. Dr. Rakeman joined the foundation virtually the end of March, but now is back in the office. You'll see him in his office today. He's been a great addition to our staff. So if you don't know him, I welcome the opportunity for you to get to know him. Andy, I'm going to turn it over to you now. Thanks for moderating today.
As Kerri mentioned, I'm still relatively new, I guess, to the foundation, having joined in March, and it's been a wonderful experience coming in and working as part of the research program, because it is such a successful program and there's so many exciting things happening. And my goal for today is to hope that everyone here leaves with that same sort of sense that this is a really exciting time for the research that the Lustgarten Foundation supports and research in pancreatic cancer in general. But what really has made my transition to the foundation most exciting and most special is getting to work with all of the scientists that we fund and we support. And two most special ones that I get to work with are Dr. Tuveson and Jaffee, who we're going to speak with today. So I'll just give a quick introduction about I know many of you will know them and know of their reputations, but of their titles and credentials, and it will have to be quick because if we went through it all, we would run through the hour pretty easily there. But starting off with Dr. Tuveson, who is the Lustgarten Foundation Chief Scientist, and in addition to that is the Director of the Cold Spring Harbor Laboratory Cancer Center, as well as running the Lustgarten Foundation dedicated lab at Cold Spring Harbor. And those of you who have been following Dave's career know that it's not just titles and accolades, but he has a long history of innovation and really driving pancreatic cancer research forward, beginning with contributions to developing the first mouse models of pancreatic cancer that are still being used today and are really the benchmark of mouse models of pancreatic cancer, to starting his own lab in the UK, where he did groundbreaking work on the stroma, or the capsule that surrounds pancreatic tumor, and really is part of what makes it so difficult to get drugs to the tumor. And I think recently is very well known by the foundation and this group, hopefully, for the work he's been doing at Cold Spring Harbor on developing organoids, which are patient-derived models of tumors that you can study in the lab and test drugs on. And you'll hear about some of that work as well as other things. So thank you, Dave, for joining us today. And Dr. Liz Jaffee, who is our Chief Medical Advisor for the foundation. And in addition to that, she's one of the rare scientists who works both in the lab and figures out the basic biology underlying diseases, and then turns those into treatments in the clinic and brings them to her own practice. And she's been a pioneer in the study of immune oncology, or using the immune system to help to combat and battle cancer, including pancreatic cancer. She's a recognized expert there. She is Deputy Director of the Comprehensive Cancer Center at Johns Hopkins, as well as a co-director of the Pancreatic Cancer Clinical Research Center at Hopkins. She's been a pioneer in, as I said, of understanding the immunology of pancreatic cancer, developing vaccines as potential treatments for pancreatic cancer, and really, as I said, driving those discoveries about the fundamentals of the immune system and how it interacts with tumors into new treatments being tested today and in the future in patients with pancreatic cancer. So welcome, Liz, and happy to have you on the call here today. So I'll run through a few questions that I thought about that I think are going to highlight how much progress we've made over this past year, although it's been a difficult year, and what's really exciting and what's important to come in 2021. But as we're talking, as Kerri mentioned, please insert questions into the Q&A function so we can come back to those at the end. But I'm going to start with you, Liz. And I think these days we always have to start out of thinking about the huge impact that's happened this year with the pandemic and with COVID. And it feels like COVID has upended every aspect of our lives, but it's really been most impactful, I think, in healthcare, with so much of the focus early on in the pandemic shifting to trying to understand COVID, and a lot of resources moved there, which has meant that it's had to adapt how we tackle these other healthcare challenges and crises like pancreatic cancer that we have. So I was hoping you could spend a few minutes at the beginning and talk about, in your experience, what the impact of COVID has been in terms of both the care and treatment of patients with pancreatic cancer, but also for the very important clinical research, and kind of what you and your colleagues and your team have had to do to overcome those challenges, and kind of the advances that have happened despite the challenging times.
Right. Thank you, Andy. So you're right. It's been extremely challenging early on, particularly on the East Coast, where we had the biggest spikes earliest. We knew very little about the virus, so we had to take significant precautions. And I think, as most people who were on the East Coast know, often the governors had basically issued these stay-at-home orders. And so this made us think, how are we going to take care of our very sick patients? And so telemedicine has become now quite a major component of what we do. And at first, we thought, well, this could hinder our ability to take care of patients. But actually, I think that it's not going to go away after COVID. I think we've learned that there are situations where it's more convenient for the patient to have these check-ins over a Zoom conversation. And even for research, clinical research, if we're only trying to see how a patient's feeling and try to get information about potential toxicities, this could be done if the patient's feeling okay through telemedicine. So telemedicine will probably go to a hybrid situation in the future, and it allows us also to reach patients who have a difficult time finding a major cancer center or being able to access a major cancer center. So that's a positive. I think we've also, for just clinical care, we've looked at different ways of trying to provide patients with treatment safely. So for instance, we have a drive-through component now at Johns Hopkins, and I think other places are doing this too, where we can administer easy treatments and patients don't have to get out of the car, with the ability to access their charts, to access the facilities pretty easily through the internet, through their phones. We've been able to make access a lot easier and make it more convenient. And we're not going to go back. Patients like this. The hardest part has been on clinical research. And it's difficult because a lot of clinical research, we don't know if it's going to benefit a patient. So we've chosen during this time, particularly in the first couple of months, to recommend as much standard of care as possible. Sometimes we had to delay surgeries if it wasn't an emergency, in order to maintain the safety of the cancer patients and also to conserve resources. It's hard to imagine why that would be, but if you think about it, many of the surgeons and the anesthesiologists in the surgical departments or the people in radiology were dealing with the COVID patients. And so this led to a reduction in standard types of surgery. So if we could give chemotherapy first, we did, as long as it wouldn't compromise the patient's treatment. Some centers didn't fully close down on clinical trials. So if you were already enrolled on a clinical trial, we maintained that. But one of the losses was in understanding how the treatment was working. So for instance, if you've ever been on a clinical trial, we often get some blood or some biopsies during the course of the treatment. And this, of course, was more difficult to do, but we were able to maintain the treatment and also monitor safety through telemedicine. The good news is that during that time, and we were down for 3 months, and I think that was kind of the typical at most cancer centers, we figured out ways so that when this spike happened again, which we all anticipated in the fall, that this would happen, we would have mechanisms in place to prevent shutting down. And so far, those mechanisms are working. And so we're very hopeful. We're back up to about 95% of what we were doing before COVID. And we're hopeful that we won't have to reduce clinical research, although again, we may have to modify what we assess, such as biopsies, if those become difficult because the COVID patients are requiring those services first. So I think overall, we had a small hit, but we were able to quickly get back up, and we're hoping to be able to continue this at this point.
Thank you so much for that. And it's great to hear that, with the innovation and ingenuity within your team and the teams all across the country, that you've been able to find ways to keep the clinical trials that were up and running. And as you said, after what was in the long scheme, a pretty short delay, be able to restart even new clinical studies. And I think we'll come back a little bit more and talk about some of these exciting new studies that are getting underway now. But Dave, now, turning it to you from more of the lab research side too, it's also been a tough year as labs have had to figure out, ways to do research in socially distant ways or keep things going. But one thing that's really struck me is that the pace of new discoveries has not seemed to slow down. And we do see a lot of new papers being published, new discoveries being talked about at the research meetings that are happening virtually now. We just had our SAB meeting a couple of weeks ago where we talked about some exciting work. As you look back over this past year, are there some advances or some discoveries that really stick out to you as kind of most exciting, that are, making you or, helping you to think about what's really going to be the strategy for pancreatic cancer research in the future?
Yes, thank you, Andy, and welcome everybody to our webinar. I want to thank you for participating in it. And I echo the words of Kerri, Kaplan, and Dr. Jaffee, that as we all know, this past year has been an adventure that we don't wish to repeat, but we did learn from it. And as Dr. Rakeman just intoned, we actually have made probably different progress over this past year compared to what we would have. And the reason is, over the past year, many scientists have continued to work on their projects. If they're not able to go into the laboratory 24/7 and only 3.5, then it turned out they would work on their computers at home, and they would analyze the data that they had accumulated over the past year and talk much more frequently to their colleagues and their team members. And so Dr. Jaffee talked about this concept of telemedicine, which is a term you all know now, telemedicine. Well, I would say this last year had telescience. The scientists were very connected. So I had a slogan for my lab. It's a Post-it note above socially distant yet scientifically connected. That's what I told my laboratory. And so I have a quite large laboratory, and they are very energetic, and they didn't want to stop anything. Practically had to imprison them to keep them from coming in on the weekends and at nights and all of this. And so they learned to work on their computers more often and to connect with their colleagues in the lab we were on a rota where you could come in one of 3 shifts. And teamwork emerged from this within my own laboratory. People helped each other finish and start experiments and interpret the results And I was very encouraged by this. And so that was just sort of in my own domain, seeing this happen. And so like Dr. Jaffee, I advise a campus here at Cold Spring Harbor Laboratory. And I think we became an example of how we could accomplish things during the pandemic. Now, the pandemic is still with us, as we all know, and I hope you're all staying safe, because we need you to stay healthy with us so we could finish the jobs we started all those years ago. And I think we can do it. Our example with our own laboratory has affected Cold Spring Harbor. I think Liz in her clinical research and laboratory at Hopkins has done the same there. And so I have seen over this past year actually different accomplishments than I would have. And I do think the spirit of collaboration is much stronger this year than it was last or the year before. And I'm hoping we keep that as we get back to the 24/7 lifestyle. And I think we can do it, at least in our area of pancreatic cancer research, where workers are much more willing to share their ideas and their approaches than some other fields, because they're all trying to help folks like you who are calling in to look for advice about this disease and look for reasons to be hopeful. And so we're hoping in the next 20 minutes to give you those reasons to be hopeful. Yeah. So I'm going to turn it back to Dr. Rakeman, who's the MC.
Yeah. So thanks, Dave. And I think, and we'll come back to some of these theories about these themes about collaboration and working through problems, because I think it's going to pervade in all of the exciting things that have happened. But maybe, you know, taking it a little bit back to the science, and this will be a question for each of you, and maybe this time we'll start with you, Dave. You know, what is it that you're most excited about thinking that's going to be upcoming in 2021? And, you know, what kind of research that, you know, is either building on some new discoveries that happened or new areas that you're going into? You know, what are you most looking forward to seeing happening in the next year?
Yeah, I mean, thank you, Andy. I see actually science for the first time in the history of our foundation begin to enter the clinic. And, you know, it cannot be understated. When we recruited Dr. Jaffee to join our foundation several years ago, it was with the aspiration that her gold standard, world-leading approach to treating patients could be married to the fundamental research that Kerri and the Lustgarten Foundation has invested in for the past 22 years. And that wasn't just, talk. It was actually our plan was to take science and present it to Liz and her colleagues so they could consider it for testing in clinical trials. And Liz will talk about how that has impacted her perspective of pancreatic cancer and actual activities that her committees have come up with. Because I see that happening, I've been able to then reflect on where are we still deficient in the field of pancreatic cancer research. And with Liz's help and the scientific advisory board's help in the foundation, we've come up with 5 areas where we think we need more work. And in fact, we've even decided that 4 of the areas we're ready to fund people in if we could find the right people. And so later today, Dr. Rakeman will be convening a board of leading authorities to select the winners in this pursuit for 10 new astronauts to fight pancreatic cancer in the areas of inflammation, which you can think of as pancreatitis, which I think happens in every patient before they become a patient. Stroma, which I've called before the oatmeal in the oatmeal cookie, the part of the tumor that makes it hard for drugs to get in and make it hard for Liz's immunotherapies to have the biggest punch. Metabolism, which metabolism is how we live, we eat, and we process food. We need that as fuel. And so as that fuel comes into our body, it makes us healthy. But if we use the fuel incorrectly, we end up with illness. And cancer is one of these illnesses. And pancreatic cancer has a very unusual metabolism. And so that's a very unusual part of this disease. And we're going to look at that. And then the 4th one is spread of the disease, or metastasis, which is a term which we use to describe the more advanced forms of pancreas cancer and studying that from a scientific standpoint. So again, back to Dr. Rakeman, I'm very excited that this next year, we will see new ideas applied to these 4 big problems, with the goal that within several years, there will be findings that Dr. Jaffee will look at with her committee as potential new ways to either treat, prevent, or detect pancreatic cancer. So that's what excites me, keeps me going. And we're hoping to be able to tell you some outcomes for that very soon. Andy.
Thanks, Dave. And maybe Liz, that's a great segue to bring it to you, as Dave set it up, of he's trying to get what's going to keep you busy, you know, in the coming years. But what maybe you know what you're excited about. You can talk about some of the new trials that are getting underway and some of the ones that you hope to see getting underway in the not too distant future.
Yeah, absolutely. What Dave said is absolutely true. There, with 20 years of funding by the Lustgarten Foundation and others, we really understand a lot of biology. So this biology needs to be translated into good therapies for patients with pancreatic cancer. And as I like to say to my people in my group, pancreatic cancer patients can't wait. We have to do it now. We should have done it yesterday if we could have. And so I'm really excited that I have this opportunity through the Lustgarten Foundation, partnering with David and Andy and Kerri and others, to be able to develop this new innovative accelerator program, which has brought together experts in pancreatic cancer clinical trials from all over the country. And so we can now very rapidly evaluate new studies, so new drugs, new combinations of drugs, and test them rapidly in patients and provide this opportunity to people all over the country with pancreatic cancer, making it convenient and also giving better access. So what have we done so far? Well, so far, we've been really actively doing this for a year. COVID did happen, but despite COVID, we were able to get 2 studies up and going. We have 2 new studies going right now. One is a chemotherapy study looking at whether we can predict who will respond to the 2 standard of care chemotherapies, and in doing so, developing organoids that David and his team can generate so we can test the next set of drugs and determine what these patients might respond to as second-line therapy. So patients are already being treated on that study. We have 2 sites open so far. A few more sites will open. And this is a collaboration even with people in Canada, so that's very exciting. The second study is an immunotherapy study. It's based on an initial study that I helped to work on with the Lustgarten Foundation. Science was developed by Doug Fearon. And this is testing a drug that targets a new immune pathway in pancreatic cancer cells. Basically, it's a pathway that is the way in which those stromal cells, the oatmeal, converses with the tumor cells to prevent the immune system from getting in. And so we can disrupt that pathway with a drug. Now, that one drug alone seemed to have some activity in the first study that we did, but we figured out a way to make it even more powerful and combine it with another drug. So we already started working on that clinical trial as well, and that clinical trial just enrolled its first patient. So even though it was held up a little bit by COVID, we're back up and going, and we're enrolling patients. So those are 2 studies, but this innovative group that I have, this accelerator program, we're not stopping there. So we want to have as many good clinical trials available as possible. And so over the past year, we've actually evaluated 10 concepts. We've looked at the science that Lustgarten is funding. We've looked at the science that's being funded at other centers, and we've looked at what's come out in publication that might be of interest. And then our committee meets once a month, and we have the investigators who develop the science talk to us, tell us about it, and we decide how we can quickly test these concepts if we think that the science is strong enough to move into patients. And so far, we've been able to review an additional 10, as I mentioned, 6 of which we now took to the second level, where we're trying to decide with outside reviewers if these are worthy of clinical development. And if so, we hope to open even all 6 over the next year if if we determine that all 6 are of value. And we're looking at different kinds of drugs, immunotherapy drugs. We're looking at drugs that target the oncogenes, such as mutated KRAS. We're looking at drugs that target stroma, again, the cells, the oatmeal cells that surround the tumor cells. So we're really trying to take this from the point of view of what has science taught us and how can we now take advantage of that science and really make progress in patients.
No, thanks, Liz. That's really exciting. I think those are some wonderful stories that show how early investments and early funding, taking risk, can lead to payouts, and why it's so important to have a foundation and like Lustgarten, that's driven by patients engaged. And just as a reminder, though, folks, if you have questions, please type them into the Q&A function. I have a couple more things I'm going to ask Liz and Dave about, maybe a little bit bigger picture, and then we'll dive into some of your specific questions. But I think, listening to the two of you talk, and one of the things I've been very struck with getting engaged with the pancreatic cancer research community is just how collaborative of a group it is, and how well people work together. We heard the example of research coming out of Doug Fearon in Cold Spring Harbor that you worked with him, Liz, to turn into a trial. Dave talked about collaborating within his lab, and that being really a model for how the whole Cold Spring Harbor is working now. Maybe if the two of you can just editorialize a little bit on the importance of collaboration and what foundations do in terms of how they fund research or how they incentivize people to work together, and why that's so important, especially with such a complex problem like pancreatic cancer.
Sure. So Andy, I'll speak first and then turn it back to Liz. So I would say from the basic science standpoint, oftentimes we spend time crystallizing what the problem is and then trying to identify people that we think will provide a solution through their investigations. Now, the discovery process is unpredictable, but we try to hedge our bets by supporting people that have been successful in the past, people that clearly know how to look for buried treasure, as it were. We have realized with time that people find the treasure, meaning they make the discoveries, much more readily when they have people to talk to, people to bounce their ideas off of. And oftentimes it allows them to avoid wasting time and focus their efforts on a more fruitful direction. And so what we have found in our foundation is that open communication amongst investigators that we support leads oftentimes for them being able to finish their projects and actually determine whether their initial ideas are correct or not. And I think the organoids is a great example of that. And so Liz mentioned this model system, which many scientists now use in the field. We're trying to determine clinically if they'll be useful, meaning will they be a petri dish that we can study cancer in, so that when a patient who's diagnosed with cancer, perhaps the physicians can be told what medicines work and what medicines don't work, so that when they go to counsel their patient, they can tell them this and reveal that. And that's the clinical trial that Liz is leading on. That's an international trial right now. It will determine whether this model system is useful. Now, we only got to this point because of collaboration, because of working together with other groups, be they groups in Holland, groups in Boston, groups, Liz's group at Johns Hopkins, groups at Memorial Sloan Kettering, groups practically at most places around the country, have worked with us to help refine those methods to make the procedure reproducible and fast. And this was something that the Lustgarten Foundation organized. It happened much more quickly than we could have if we had gone to the government, for example, to ask for resources. The government is a terrific supporter of medical research. Oftentimes they need a level of proof that you're on the right path, that they don't touch things that are extremely risky. And what we do is we try to de-risk the risky things by getting people to share the risk. And that's where the collaboration becomes so important. And so from the basic science side, we try to make discoveries more quickly than other modalities. And we've done so by actually nurturing this field of people who are comfortable enough to talk to each other compare notes, cheer each other on when they need to, argue with each other when they need to, all in efforts to work as one team to get it to the point where we have something worthwhile to talk to Liz and her colleagues in the clinical arena.
So I guess I'll just add to what Dave said. I don't want to spend a lot of time since there are a lot of questions already coming up, but I think how collaboration has really jump-started and accelerated our process is that this monthly meeting we have where investigators bring their science, they don't know how to develop a clinical trial. So we have a team of experts who really help them design the study, design the right things to measure to show that the drug is working, and decide what patient population would benefit most from that particular drug. So I think it's very important because we teach basic scientists or early career scientists how to really take their discoveries and turn it into something that can be tested well in patients and potentially have an effect in patients.
Thanks, Liz. And now, and I think those are great examples. And I think even as some of these questions come up, you're going to hear these common themes about collaboration and working together and taking risks. And a number of questions coming up in the chat here about earlier detection. And I know that this is something that's very important to a lot of people, and people asking questions about using advanced imaging methods to detect pancreatic cancer earlier, or are there blood tests being developed? And is this something that's only going to apply with people who have active disease, or is it things that we're going to help to even screen people who are at high risk, either because of family or otherwise? And now I know this is an area where over the past year, there's been a lot of exciting developments, both scientifically and business-wise. So I don't know, Liz or Dave, if one of you wants to take a stab and give an overview of some of the exciting things that have happened in detection of pancreatic cancer.
So I will just say some general things, and I will turn it to Liz. What Andy said is correct. Both blood tests and imaging tests are currently being evaluated very carefully by the Lustgarten Foundation. Two big projects are being supported. The blood test project is called Cancer Seek, where Dr. Bert Vogelstein, one of our dedicated lab leaders, developed this approach, and a large series recently showed that this approach could be useful for detecting certain cancers in 55 to 65-year-old women, a study out of Geisinger Medical Center. So we are currently in discussions with the Cancer Seek supporters about applying that to pancreatic cancer by evaluating families that are at increased risk for pancreas cancer, as well as others who don't necessarily have a family but have other features about them. So that is a very important project for us. And the other area that Andy mentioned, and some of you have asked questions about, is imaging. Again, Dr. Vogelstein, he works 24/7, has a different project called Felix, where devising better scanning methods to find small tumors has been the goal of that. The work is very provocative and exciting. It's not quite finished, but the whole point of that is to use artificial intelligence and machine learning to refine the analysis of scans so that small tumors can be seen. We're hopeful that then this would lead the patients to be referred to Dr. Jaffee and her colleagues. But her perspective on this will be quite interesting because this is also an area that she works in, so I'll turn it to her.
Yeah, as some of the questions have pointed out, it's been a real challenge to conduct early detection, and not because people haven't tried very hard, but I think that right now, much of what we're trying to do is just what Dave says. We're trying to support imaging. We're trying to support new gene technologies, genetic technologies that can detect the earliest molecular changes when a normal cell starts to transform to a cancer cell. And many of you have heard of mutated KRAS. That's one of them, but there may be, when you combine a number of them, it may be even more sensitive for detection. And that's what Bert Vogelstein has been trying to do. And there's been several groups trying to do this in different ways, different technologies. So although we don't have something yet, we do believe that we should hopefully have something, at some point. There's so much investment that everyone realizes how important this is. And again, one of the areas that we talked about is imaging, and we also had a nice discussion about new imaging technologies that might be used to detect even very small tumors that you can't see with the naked eye. So I'm hopeful that there will be new clinically applicable imaging and also blood tests at some point.
And just to add, and I don't know if folks follow the financial news, some of you may have seen this, but the CancerSeek that we've talked a little bit about, which is a project that the foundation funded from its very earliest days as a sort of a biological discovery project. And then Hopkins was able to spin out into a company called Thrive that's been further developing that. But we know, in order for something to hit to patients, it not only needs to go through the research and the clinical testing, but needs the business side behind it. And that company now has just done a very large deal with another diagnostics company that's hopefully going to really help to push that technology over the finish line if it is something that proves to be effective in the end. And again, this is a technology that has the potential to detect not only pancreatic cancer earlier, but other forms of cancer too. So it could hopefully be, very impactful to society as a whole, and certainly to our patient population. Some other question for you, Liz. People asking, I know there's been a lot of interest in immunotherapies, and immunotherapies are really, revolutionizing not only how we think about cancer, but how we think about treating a lot of diseases. But I think not everyone understands fully how they work, because maybe you could just back up a little bit. And what do we mean when we say an immunotherapy? How is that going to work? How does that help us to treat cancers better?
Okay. So immunotherapy, when we talk about immunotherapy right now, we're talking about two types that have been approved for other types of cancers. They're the immune checkpoint inhibitors. Those are antibodies that actually block stop signals or inhibitory signals on immune cells. The T cells are the best immune cells right now that we have been trying to get activated. So these immune cells come into the tumor and the tumor stops them from functioning. So these antibodies prevent the tumor from doing that by targeting particular molecules on the T cell and on the tumor that normally would cause the T cell to stop working. And so those are immune checkpoint inhibitors. You've heard about them, anti-PD-1. Many different companies have them, so I'm not going to give you the commercial names, but it targets a molecule that is called PD-1 and PD-L1. Those are the two proteins that the tumor and the T cell use to crosstalk to say, stop, don't be activated. Well, why would that even happen? I mean, you got to ask yourself, why can't the T cell just get in there and kill the tumor? Because it does see that the tumor is different from the normal cells. However, the normal cells around there, that's their mechanism of saying, hey, we don't want you to kill normal cells. So the tumor basically, tumor cells learn to adapt. And so now they're adapting this mechanism to prevent the normal cells from being killed. So it's a way that they escape the immune system. So this is one way to bypass that escape. Now in pancreatic cancer, unfortunately, the T cells never really know the cancer's there. Pancreatic cancers, they just don't, they're not very visible to the immune system from the start. Some cancers are very visible, such as lung cancer and skin cancers like melanoma, not pancreatic cancer. So one of the hypotheses is that you have to give some sort of treatment to alert the immune system that the cancer exists. And so one way to do that is to use vaccines, not for prevention, but to use vaccines that can educate the immune system to recognize the proteins that are different on the cancer compared to the normal tissue. Another way of doing it, sometimes chemotherapy could do that, or radiation therapy. So we're trying different ways to alert the immune system to the existence of the cancer. The other way, the other form of immunotherapy that has been approved is something called CAR T cells. Those have been approved for leukemias currently. And those are basically T cells, the T cells like in your body, except they're taken out of a person's body, and then they're engineered to express certain proteins that can now traffic to the tumor and attack the tumor. So they're attacking those proteins because they've been engineered to. So instead of using a vaccine to get them induced in the patient, we're taking the T cells out, engineering them, and putting them back in. So those are the two types of therapy that we have. In pancreatic cancer, immunotherapy is experimental for the most part. There is a small percentage of patients that have typically a genetic form of pancreatic cancer, about 1% to 2% at most. And those patients will respond to anti-PD-1. And so that is some progress, but it's not anywhere near where we want to be. So we've been trying to build on that progress.
And I know that that's been a lot of focus of the clinical accelerator initiative is how do we get around these mechanisms that make the pancreatic tumor so invisible to the immune system? And even it's the basis of the study you talked about with Clark-Sephora earlier that comes out of the work with Cold Spring Harbor, correct? So another question coming up, and maybe this is a good one for you, Dave, to address. It speaks to what are the environment, what do we know about environmental triggers? The question is specifically around foods and things that we eat. You talked about metabolism, but I know smoking and other things. And, you know, even if we don't know very specifically about, you know, certain triggers, maybe you can also talk a little bit about what we've learned about how different triggers impact inflammation in the pancreas and why that ties in with this new grant mechanism that we're looking to fund next year.
Sure. Well, pancreatic cancer is a type of cancer that's not as common as others, but it is becoming slightly more common over time. And we have learned by studying populations that there are associations that one can build between the exposures that a patient has or the family the patient has to the disease. And so, for example, we know that pancreatic cancer is a little bit more common in men than women, but it's barely. It is more common, though, in African Americans than it is in Caucasians or European ancestry. And the outcomes in pancreas cancer are a little bit worse in African Americans than Europeans. Similarly, in Hispanics, the outcomes are a little bit worse than it are in Caucasians. Now, we have been thinking about that. And in fact, we have recently worked with AACR, American Association for Cancer Research, to fund the first fellowships in understanding these relationships. As you are all well aware, we recently lost Supreme Court Justice Ruth Bader Ginsburg to pancreatic cancer. Now, she had pancreatic cancer for a long time. She fought like a warrior. She had terrific doctors, but eventually the cancer won. And the foundation has decided to start an RBG fellowship in pancreatic cancer research with AACR, where we will support women physician scientists to work on this disease. Similarly, we lost Congressman recently, John Lewis, to this disease, who is a well-known African-American congressman. And as I said before, African-Americans have a higher incidence and worse outcomes from pancreatic cancer. And we will fund a fellowship in this, trying to attract into the field people of color to work on this disease with us. Now, we think there's something underneath this that's either related to the genetics of Hispanics and Africans compared to Europeans, or something else. And the something else could be habits, could be, as Andy said, tobacco. Tobacco is a well-known risk factor for developing pancreatic cancer. Or it could be other things which stimulate inflammation of the pancreas. This could be food, it could be microbiome, which are bacteria that live in your intestines. It could be obesity, which we know is a link to pancreatic cancer. All of these things collectively can promote inflammation of the pancreas. Metabolism. Metabolism, again, is how you run your car. You put gas in the car and oil. And healthy people use gas and oil a certain way. In cancer patients, they use those fuel sources differently. And we know in pancreas cancer, the cell eats food differently. And whether that's related to the food a person actually eats or not is something we still don't know, but is an active area of research. And so we are pursuing that because that allows you then to modulate your approach to this disease. One of the questions you all had is, could I change my risk profile by inserting certain foods and avoiding other foods? I would have to say, as of now, all I can say is try to avoid being obese, try to exercise regularly, try to have a balanced diet as just general advice. There certainly has been advice from physicians about limiting your intake of red meat, particularly barbecued red meat, because they have certain carcinogens in them. But there's not a magic formula yet for how you can avoid pancreas cancer by a certain diet versus not. Other than avoiding tobacco, excess alcohol, things that cause obesity, or excess sweet food, things high in sugar. Those would be the general recommendations we have. But we hope that the people we recruit into studying this with us will come up with some answers for your question. And that's what we're going after with the science we're funding right now.
Thanks, Dave. And as I probably could have predicted from the outset, we're going to get more questions that we're going to have time to answer here. So I think we're only going to be able to do one more, so we want to be respectful of everyone's time and finish up on time here. And seeing one here that I think really can sum up a lot of what we talked about that maybe is a good one. And it's a very simple question. It's, why do you rarely recommend removal of a cancerous pancreas? And I think, it's going to come back to what are a lot of the challenges that we need to overcome in pancreas cancer. We've already talked about some of them. The immune system doesn't like to see the tumors. It's in a tough place. But maybe, Liz, you can start just beginning from the kind of clinical perspective of why is it often that a diagnosis is so hard and surgery often isn't on the table. That sets up basically all of the work that we're doing to try to get to newer and better therapies.
Yeah, it's a really important question. So unfortunately, by the time we detect pancreatic cancer, 70% of patients are no longer surgical candidates. And the reason is that the tumor has either grown so much that it's constricting blood vessels and nerves, and you just can't, these are major blood vessels and nerves, you can't just remove them. They're part of life. So that's one problem. And then the second problem is about 50%, so 70% total, 20% are going to be in that situation. The other 50%, unfortunately, have tumors in other places in their abdomen, like their liver or their peritoneum, the lining around the abdomen. And so if you remove the tumor, the recovery rate could be, 4 to 5, 6 weeks, and it's not going to help the lesions that escaped, and it's going to delay treatment of those lesions that escaped, those cells, the tumor cells that escaped. And so the best treatment is really a systemic treatment, which would be to start chemotherapy. Now, our chemotherapies have gotten better, the first-line chemotherapies, and there's two types. One is the FOLFIRINOX, which is a combination of a number of different drugs, and the other is gemcitabine given with Abraxane. And this is the reason we're testing these two, to really better understand which patients will benefit from which of these treatments. But we know that there's a small group of patients who do very well for years on these treatments if they can handle them. And sometimes you could shrink the tumors enough that the metastasis, most of them go away. And there are circumstances now where, depending on the situation, if they've been stable for a long time, we can go in, remove the primary tumor, and remove a liver lesion or a lung lesion. And we've done that, and patients have gotten good benefit out of that. But again, that's still not the usual scenario. But as our treatments get better, we'll be able to do that. Now, the problem is that we can't detect early, and that's really the issue. And that's why Lustgarten Foundation has put a lot of effort at early detection methodologies. And I think one of the questions was, which one's better, the Felix or the cancer-seq, and we still don't know that answer, but we're hoping that both together might be even better than just one. And we have to be realistic. We're probably going to need more than one way of monitoring people at high risk for pancreatic cancer.
Thank you, Liz. And I think, so I think this has been a wonderful discussion. I mean, I think if I started out, I said my goal was everyone here leaves learning something new and excited about what's to come. I hope everybody took away something from there. I think you heard a lot about advances that we're having really across all the different priority areas for the foundation, understanding the basics of the disease, developing new drugs and testing in the clinic, finding ways to detect things earlier, and then really tailoring that treatment back to the individual's needs. And I think what Dave said in the beginning, if I had to pick what I'm most excited about, it's something Dave said early on, that across all the different areas that we've picked out as high priority, we're now testing new approaches in the clinic in patients. It's not just an academic exercise in the lab or curing more animals. Really, at the point where we're putting our money where our mouth is and testing if these are going to be things that work out for patients today and patients in the future. And that, to me, is why it is such an exciting time, both for the progress, but also for the need to keep moving forward as fast as possible. So I'm going to stop there, other than just to thank Dave and Liz for all the time you put in today, but the time that you put in 364 other days of the year to help us get through this, and for making this such a wonderful discussion. And with that, I'll turn it back to Kerri, I think, who will help to close us out.
So I really want to thank Andy for doing such a great job of moderating, and for Drs. Tuveson and Jaffee for, really sort of summing up and answering so many questions. I know in the chat, everyone really wants to know when there will be a blood test, and we are working very hard to make sure that if there is blood test, that it is the right test. But, you should know the goal is sometime soon that for those at high risk, which many people on this call, will fall into that category, that the next clinical trial will be for people at high risk. And you can be assured that, when that day comes, we know where to find you, and we will reach out. And, we're working very hard to make that day come, very soon. So, it's really, I think, early detection, and then of course, better therapies. That's really our priorities. And I'm really excited that, these things are moving now towards the clinic. And, when we have this next year, we're going to tell you about which trials are actually working, and what has been successful, and what we've learned, and what's going to be next. So I just want to conclude by letting you know just a couple of things that will be coming up at the end of the year. You'll be getting emails from us. We will have a big year-end appeal, and thanks to the Johnson Charitable Trust, at the request of Sandy and Steve Waters, Tara and James Waters, and Anne and John Enberg, and the Burns Schwartz Family Foundation, will be announcing another year-end match. So donations of up to $125,000 will be matched for year-end. We know that's something that you all want to hear about and get excited about. If you go on our website, you'll see also on our homepage, we've launched our virtual 2020 Holiday Bash fundraising campaign that will run through the month of December. And we will continue to be walking virtually, at least through the spring. We'll keep our website updated. We hope that later in 2021, we'll maybe be able to go back to in-person events, but if not, we'll still be spreading hope from home, including encouraging people to continue their popular Facebook fundraisers. So, a lot more to come this year. And I want to give a couple of special shout outs. You heard about some exciting programs today. So the first clinical trial in the Clinical Accelerator Initiative is actually happening because of funding from the Stephen and Nancy Graham Philanthropic Fund. So we're really excited that they've helped us launch this, and will let us do that many more programs. And you've heard about the organoids, which has been, you know, a baby, sort of a baby of the Lustgarten Foundation, and brought to us as soon as David Tuveson, you know, opens his dedicated lab. And that's been funded by the Gail D. Coleman and Kenneth M. Brutel Organoids for Personal Therapy grant. So, you know, thank you to all of our donors, but I wanted to just point out those couple of grants that are really moving forward. And I want to give a final thank you to Ipsen for sponsoring the webinar, and really to all of you for spending this hour with us. You know, we hope today has proved to be, you know, giving you some good information and giving you, we feel very hopeful. We feel like there's so much momentum in this disease, and we are in this fight with you with the same worries, but we feel so hopeful that we're really turning a corner and a lot of progress is being made. So I just want to wish everybody a safe and healthy and happy holiday season and New Year. Thank you again to our panelists, and that's it. Thanks for spending the hour with us. Bye-bye.